Now let's look at the process of translation, how the language of nucleotides is converted or translated into the language of amino acids. We have seen the structure of ribosomes. In prokaryotes, the ribosomal RNA of the small ribosomal subunit first binds to complementary ribosome binding site upstream of the start codon. What I mean by that is the small subunit will bind the messenger RNA. There is some extra nucleotides on the messenger RNA. These are the extra nucleotides you can see. The start of codon is right here, and this is where the protein will start making. So small subunit binds this specific region, the region of this RNA or DNA, which is towards the 5' end of region of interest, and here our region of interest is start codon. It is called upstream. The region which is towards the 3' end would be called the downstream region. These are specific sequences on all messenger RNAs that basically have the information that allows the small subunit to bind it and initiate the process. And this, in case of prokaryotes, that sequence has a name also called Shine-Dalgarno sequence. The anticodon, as we have said that we looked at the genetic code and we said the start codon is the codon which basically is coding for methionine. So the first codon is always for specifying methionine. So tRNA, which is carrying methionine, will come and attach to the start site. So when this happens, here you can see the attachment of tRNA with methionine at the start site. So when that happens, the large unit comes in and it also engages with the complex. Here I would like to mention that methionine is always the first amino acid as a rule, but it is not first amino acid on the initial protein, but methionine can also be cleaved off in the mature protein. So it is not necessary that all proteins start with methionine. So small subunit of ribosome, which has the ribosomal RNA, this basically stabilizes the hydrogen bonding between the transfer RNA and the messenger RNA. So this ribosomal RNA present here is stabilizing this hydrogen bonding right here between the codon and the anticodon. The large subunit, it comes in, it gets help from facilitation from initiation factors that allow this large subunit to come in, and the site the large subunit lands on this complex is that P site is where the transfer RNA containing methionine is located at the P site of this large subunit at this point. So let's keep moving. This is the initiation. So elongation. Now we have initiated the process of protein synthesis. A charged tRNA with anticodon to the next codon binds the A site. Here you can see, here is the A site. This is the anticodon on the tRNA, and it is complementary to codon right here on this messenger RNA. It will come in and it will bind at the A site. It is also assisted by elongation factors. Then see what is happening here is the large subunit is, remember this was a high energy bond between the amino acid and the tRNA. This large subunit first breaks this bond, which is basically carboxy terminal of this amino acid bound to the 3' hydroxyl of the 3' carbon. This bond is broken and the bond between the two amino acids is formed, which is, we know, the peptide bond. So the first amino acid will be the N amino acid or the amino terminus of a protein. So this methionine is now transferred to the next amino acid, and this process keeps on repeating itself till the process is terminated. Let's see how the termination takes place. When the stop codon enters the A site, elongation is terminated. this codon does not have any corresponding tRNA. This codon basically binds another peptide, another factor that allows this final protein product or the polypeptide to be cleaved off and released in the cytoplasm. So the next issue comes as how the protein folds on itself. The protein folds, the folding information of a protein is contained in the amino acid sequence. We talked about this when we were talking about the structure of proteins. Here we can see an animation. The small subunit of ribosome comes in. It initially binds the Shine-Dalgarno sequence, whereas the x-axis are, it moves to the start region. The initiation complex, first tRNA comes and joins, and later the large subunit. The next codon is also read. Appropriate relevant tRNA comes in, and the amino acid is transferred to the methionine is transferred to the arginine. This process keeps on repeating. The amino acids are getting attached, and the ribosome is moving along the messenger RNA. So now this bond will be glycine, and the tRNA will be broken, and glycine-serine bond will be formed, as you can see on the screen. So this is how a protein chain is growing. Addition, sequentially adding amino acids one by one, reading the codon, and codons are specifying specific interacts with specific anticodons on the tRNAs, and the fidelity of the system is maintained. The stop codon, when it comes in, as we said, there are no tRNAs which do that. There's other factors. They come and release the system. They release the polypeptide, and the ribosomes, they fall apart. So both the subunits, the small and the large subunit, they will fall apart. Messenger RNA will be released. The protein will be released. Where that protein is going to go, we know the eukaryotic cells are compartmentalized. They're membrane-bound organelles. So the information is coded with the address or the location of protein is also part of that protein molecule, and how that protein is going to fold is also encoded in the sequence of the amino acids.